All right. Hi, everyone. I think we're going to get started. So my name is Jamie. I am a marine biologist and ocean educator here at the National Museum of History. And we have a really unique experience today to talk to some coral aquarists located in Galveston, Texas. So this lab is in Texas, and we're going to learn all about kind of what it takes to grow corals in a lab. So before we do that, though, I'm going to give you a little bit of background on why this project is even happening and what's going on. So we're going to watch a really short video. So the deep sea is the largest habitat on Earth and is home to an incredible diversity of life, including deep sea coral reefs, like the one you're seeing here. In the Gulf of Mexico, once you pass about 150 feet below the surface, less than 10% of the light from the surface reaches the bottom. But still, a really vibrant ecosystem thrives full of corals, sponges, fish, and invertebrates, like the ones you guys are seeing on this screen. Despite how remote these ecosystems are, humans can still have an impact on them. In 2010, the Deepwater Horizon oil spill released 134 million gallons of oil into the Gulf of Mexico off the coast of Louisiana, even reaching the deep sea ecosystems. 770 square miles of deep sea floor habitat were impacted by the spill, like the ones you're seeing on the screen now. To help restore these ecosystems from the spill, even 13 years later, restoration work is taking place right now in the Gulf of Mexico. These restoration projects include many different parts, like mapping the sea floor with things like ROVs, like you see there, to learn and document these ecosystems and all the things that live there. This restoration work also includes growing deep sea corals in aquaria. So something that we're going to do today is talk about these corals. These are corals that are grown in aquariums. This is footage from the aquariums. And we're going to see these corals today up close. So today we're going to be talking with people from NOAA's Galveston Coral Lab. It's located in Galveston, Texas. And we are going to be seeing what it is like to feed these corals and what it's like to keep them alive. So this is footage now from the Coral Lab. And we are going to talk to NOAA fisheries scientist, Ben, and he's going to tell us about these corals. So hi, Ben.
Hi, Jamie. My name is Ben Higgins, and I'm a research fishery biologist here at NOAA Fisheries, Galveston. I've been here for 32 years. I spent 30 years raising sea turtles for the federal government before I switched over to raising corals. And quite honestly, I'm not much more than a glorified plumber designing special systems for aquatic animals. But today we're going to look at some of the food that we feed these corals. And right behind me here, these are, we're hatching brine shrimp. Actually take freeze-dried eggs, and we hatch them in these hatchery dishes. And once they hatch, they end up in the center here because they come to the light. We put them in these special cones, and we gut load them, or we enhance the nutrition of the brine shrimp by adding different compounds like this RG plus, algae substitute, and Celcon, which has got lipids and micronutrients in it, makes the brine shrimp more nutritious for the corals. We also have a variety of powdered coral foods, freeze-dried powdered foods, reefroids, bunch of phytoplankton and zooplankton that have been ground up into a fine powder because a lot of these corals have very small polyps. And we end up with this coral gumbo. And we actually target feed the corals with this coral gumbo with a pipette. And now we're going to look at some actually these are live newly hatched brine shrimp on the screen here that are underneath the microscope so you can see them. And then we're going to move over here. Shannon, one of our aquarists, is going to actually show you how we feed the corals. We feed them twice a day. We feed them at 8:30 in the morning, and then again at 1:30 in the afternoon. Right now, Shannon is feeding some of the Swiftia corals. These are the ones that are the most fun to feed because they have large polyps, and each one of those tentacles, once it senses food, will grab the food and pull it into its central mouth. These are octocorals, so they all have eight tentacles with feathery edges on them, and they have a central mouth. And if anybody remembers the game operation. In that game, if you touch the sides of something, a buzzer goes off. It's the same thing with these corals. If you bump them, they all retract. The whole colony retracts, and then you have to start over again. You got to come back in a few minutes once they open up again to feed them. The corals will actually take in as much food as they can at a time, and when they're full, they'll actually keep the tentacles retracted. So you can actually tell if you've given them enough food when the tentacles stay in for a little bit of time, sort of swell up and push the food down and digest it.
These guys are so fun. I love watching them. I love watching their little tentacles retract in when they find food. Can you tell us a little bit more about what corals we're looking at that are getting fed?
Sure, we've got 2 species in this tank. We've got 4 species in total in the coral lab right now. The orange one is the Swiftia excerta, and we also have Mauricia pendula, which we're going to feed next, which is this white coral. I'm trying to stay between the camera and I don't want to walk in front of the microphone. If I go right here, I can see what's going on. So now she's feeding the Mauricia. The Mauricia has much smaller polyps, so it actually can't eat those newly hatched brine shrimp. They're just too big for it. But what it selects out of the coral gumbo, the smaller stuff like the reefroids and the golden pearls. But also, again, they're really sensitive. You just bump it just a little bit, or you push a little too hard with the water pressure, and the whole colony will pull inside. And now she's feeding some of the babies that we had hatch here in captivity. These ones were spawned, they're a little over a year ago now. These are Swiftia babies. Some of them have multiple polyps. They're old enough and large enough now that they can eat just about everything that we feed the larger corals.
So these guys that she's feeding now, they were born in the aquarium, right? They were born in your lab, and
That's
now
right.
they're growing up.
Yes, we have both. The colonies are either male or female, and we actually had sexual reproduction where we had the females releasing eggs into the water and the males release sperm into the water, and it mixes together in the water, and it produces a mobile larva that sort of floats around for a couple days, and then it starts crawling on the bottom. And eventually the larva actually then settles out on some hard substrate and then goes through a metamorphosis and turns into a polyp, and then it starts looking just like the parent. And we've had that happen with 2 of our species, not the Mauricia. If it happened with the Mauricia, we just didn't see it because the eggs would be too small. We do know which ones are male and females. Part of that's because when they were collected on the vessel, they actually took a small snippet from each one of the corals and sent them out for histology. So they actually looked at them under a microscope to see whether they had eggs or developing sperm, or if they were reproductively non-active. So we know our little guys here, these little ones in front here are all males, and some of the big ones in the back are females. And we've actually had them spawn in 2022, 2023 and 2022 and 2023. Keep forgetting we're in a new year already.
Me too. I'm still dating 2023. So can you talk a little bit about, these are really cool corals and you're growing them. Can you talk about a little bit why it's important that you're growing them in aquariums and why these resources are going towards these corals?
I sure can. It's very hard to study these animals in the wild. You have to have expensive ships and expensive ROVs to go down and study them. You only have a little bit of bottom time, but if we were able to bring them into the lab, it gives us an opportunity to study them much closer. What we know about these corals literally fits in my hand. What we don't know fills the rest of this room. We're almost learning something every day about them. We had no idea how long the larval cycle was. We had no idea how long it took for an egg to fertilize and divide. So, we're learning all sorts of things about these corals, and those are going to fill knowledge gaps that we don't have right now. There's no way that we can protect these animals in the wild if we don't have a good handle on what their microenvironment is. We need to know what their pH is and their temperature and what they're tolerant of. We don't know if they're sensitive to temperature change or pH change. And, we're learning something about them every day.
This is amazing. I think we're ready to take some questions from the audience if you are ready to answer some.
I'm ready. Bring them on.
Perfect. So, earlier earlier when Shannon was feeding the corals, she was wearing a glove. She was wearing a super long glove. Why does she need to wear a glove when you feed them?
Well, it's for a couple of reasons. One is it's biosecurity. Everything that goes in the tank, we want to make sure it's clean and it only goes in that tank. So I don't know if you can see behind me here, there's a rack of gloves. So for each tank, there's a rack of gloves. So when we move from tank to tank, we only use this glove in this tank. The other thing is, if you've got any lotions or creams or anything on your skin, that's going to end up in the water. And we're introducing something foreign into an environment which we know shouldn't be there. So it's all about keeping things clean. And another reason too is these are mesophotic corals, so we're keeping them in 73-degree water. But when we get our deep water corals, when they're kept at 40 degrees, you're going to really want that glove.
Yeah, definitely. I don't know that I want to stick my hand in 40-degree water immediately.
Not for very long. Without a glove, you won't.
Yeah. So I've got a question. We've got someone who's a sociologist in the audience, and they're wondering how this work might impact humans, or how we think about, when we're doing this research, how we think about how these corals impact humans, or how we impact the corals as humans.
Can we come back to that question? Give me a chance to think about it. I mean, everything we do.
Ways?
I mean, everybody's probably heard about coral bleaching. Everybody's probably heard about ocean acidification. Well, most of that we're seeing in shallow water stony corals, which most people, that's what you see. If you think about corals and coral reefs, reef building corals, these large coral reefs, they're near the surface, the sun is shining on them. There's algae that lives in the tissues of those corals and it produces energy for those corals. These corals don't have that algae in them, so they have to catch their own food. They live at deeper water depths. They may or may not be more isolated from things like ocean acidification and temperature change, which is now impacting shallow water coral reefs. Some of the animals that we think about, I mean, let's say you like to eat Chilean sea bass, for instance. Some of these corals live in forests at deep, deep depths and end up being sort of nurseries for smaller fish that may end up being on your table, several years later. So there's always a connection between humans and just about every other animal on the planet.
Yeah, we like to say in the ocean hall that it's all one system. We're all one ocean, and that is exactly what you're talking about there. That what we do here on the surface is going to affect all parts of the ocean,
all the way to the bottom. Yep.
Exactly. So, in the tanks, all the corals, they're on a little base with a little number on them. One of our guests asked, "What is the number on the bottom of the corals for?"
That helps us keep track of them. The number is actually the colony number. So in a lot of this tank, you'll see a lot of, there's 3-1, 3-5, 3-4. Those are all fragments from one colony that was collected as colony 3 on this particular cruise, the Mercy 1 cruise, which was on the flower gardens back in July of 2022. So wow. every time we take a branch off of a coral and put it into a new base, it just becomes 3-whatever. So we've got numbers in here that correlate with the samples that were taken by the ROV at that
time. Someone also asked, so these corals, are they one organism, or is it more than one living together?
It's a colony. So each one of these fragments is its own colony. They're all connected by a mesentery tissue that goes along the core structure. These are referred to as soft corals. They have a protein-like structure that's made out of a protein called gorgonin. It's actually, they're very flexible, and the live tissue is growing on top of that protein structure, and one polyp on the end here could theoretically send nutrition down to a polyp at the base and vice versa. So it's a colony. It's one animal, but it's made up of a whole bunch of different polyps. It's a colonial organism.
Very cool. That's what makes them unique. It's fun. So, after having these corals in the lab for a long time, is there any one moment, or are there any fun things you guys have discovered or figured out? Was it the moment when everything was spawning and you got little babies?
Oh, we actually had one of our coral, maybe two of our corals spawn on exactly the same day a year apart. And that was
Wow.
pretty cool because we were expecting them to spawn around the first moon in October. So that's when we spent a lot of time sitting in front of the tank with the water flow, very low, because when the eggs come out, they start to move around if there's a lot of water flow. And we start really looking for spawning around that first moon in October. And like I said, we've had the exact same coral spawn exactly to the date. It's like, October 20th of 2022, October 20th of 2023.
Wow.
That's pretty cool because especially in this route, there are no lights. There's no moon, there's no sunlight, everything, it's artificial light, and we pretty much keep them in the dark if we're not in here working with them. So they must have some sort of internal clock to be able to program that.
How do they know? I can barely tell the time if I don't have my iPhone. So we had one great question from a guest. They said they've heard of like metal structures that can be put into areas that can help stimulate coral growth. Are you guys doing some of the same stuff here? Like artificial structures, can they be placed in the deep sea?
Yeah, we're going to be doing things like outplanting racks. We actually have been working with putting things called landers down with different materials on those landers to see when the corals spawn, what they prefer, and if they like a certain material better than the other, then we can concentrate on using that sort of material to seed areas in hopes that more corals will attach in those areas. But in terms of metal racks, no, we're really more interested in things that are more closer to the biology than metal or steel. You may be thinking about, like things like oil rigs in the Gulf of Mexico will grow sponges and corals and things on them. And sometimes they leave them in place to create artificial reefs, but we're not trying to create artificial reefs with steel, but we may use other materials that are more biologically close to what they would be encountered in the deep sea environment. So yes, we're working with that. And one day we hope to be able to put these corals back, and we hope to put more back than we took.
Amazing. Really cool. So if there are no more questions, I think we're out of questions. I want to give you one more round of applause for helping us and talking with us today. Thank you so much. I even learned something today. Thank you for talking with us, Ben.
Anytime.
All right, bye. Thank you so much.
Sure. You guys want to see my favorite coral?
Oh, yeah. Wait. We're seeing a cool coral. Bonus footage.
Yeah, this is called Placa gorgia. And I guess it was a huge colony, like a sea fan, that was broken up for the three different government labs or 4 labs that are raising the species right now. But it's just really pretty.
Oh, wow. It's like one color at the bottom and another color at the top.
Yeah, it's real pretty. And unfortunately for us, it did spawn in captivity, but we only took one colony. It's all female. So we need to find a male Placa gorgia for our collection this next year.
It's beautiful. Thank you for sharing that with us.
Sure. Love to share anytime. Bye.
Well, thank you everybody for coming. If you have any extra questions, you can come ask me.